Okay, so Lucina did her PhD in cognitive neuroscience at UCLA. So you can see her.
Yeah.
She then did a postdoc at NYU. She was on the faculty both at Stanford and the University of Miami for a number of years. And then she triumphantly returned to UCLA a couple of years ago, where she's a professor now. She heads the Brain Connectivity and Cognition Laboratory and has made many contributions to the area of resting state functional connectivity and using and studying the functional networks that underlie social cognition and executive functions. She has received the Young Investigator Award from OHBM a few years back. And today she'll be talking to us about her recent efforts in putting some order in the chaos of nomenclature of functional networks. So please join me in welcoming Lucina to the Martinos Center.
All right. Thanks very much. There's already a question in the chat. Okay, great. So yeah, thanks, Anastasia, so much for giving me a chance to come here and very informally talk to you today and give a very short overview of some of the things we're doing since it's summer and nobody has too long of an attention span. So basically, we have been trying to think about a way to make sense of what we call large-scale functional brain networks in a way to improve communication across different fields that use human neuroimaging to understand the structure and function of the brain. Let's make sure that this comes back on. I think it's very finicky. I think so. Yeah.
The online audiences, there's no for online audience.
Okay. There it is. Okay. All right, it's fine. Yeah, it's fine now. But it won't move the slides forward. Well, okay. Now it's okay. So this is good. I'll just not touch anything from now on. All right. So this is just one of those fun figures of resting state fMRI showing you these slow, spontaneous fluctuations in the brain. I think for a long time we assumed that this was just kind of random activity and sort of noise and not very interesting. But of course, now we know that there's a lot of structure here. And in fact, you can see if you look around and let it run for a few iterations, you'll see networks that you might recognize, like a language system or visual networks or frontal parietal networks. So all of these things are kind of constantly going on. There's so much spontaneous activity that's coherent in the low frequency range. And that's what we are really taking advantage of when we do any kind of resting state functional connectivity analysis. And so what we're going to do now is actually a game, which I think will illustrate very much why we're starting this process of trying to build a taxonomy of these large-scale brain networks. So the game is to name the network. Okay, so if somebody came to you and showed you these maps and just you're sitting in the lab and they want to say, okay, what should I call this? What are we going to name this network? What would you say? Visual? Anybody have a different name? Visual, okay. So this one seems like you can have some sort of good agreement in the audience at least. So we'll go to the next one. This game gets more interesting. We'll see. Okay, what about this one? I heard three different things. Wait, no, I maybe I just can't hear.
I just said DMN.
Oh, DMN. Okay. Anyone else? Everyone's pretty happy with that label. Okay, so that was actually pretty easy, right? So it shouldn't be that hard for us to build up some kind of consensus because we can agree. It's when you see an occipital activation, you can sort of say that's a visual network. When you see this kind of medial prefrontal and precuneus and maybe some lateral parietal, you feel pretty comfortable calling it default mode network. What about this one? So we had one vote for salience. What else? Okay. Any other votes? People are thinking very hard. Okay, well, we can't linger too long on it because the projector will go away. Okay, hold on. Come back. Okay. So keep in mind whatever you think this network is, and then what about this one? Wow, really nobody, nobody ventures to name this one. So I think that's where we come in and say, okay, well, how are we even deciding in the first place? Like the first two, why were those easy, and why are these two hard? And what are we even judging them against? Like, do we even have a standard for, okay, this is what we would call visual, this is what we would call default, what would we call this one? It's kind of one of those like tests like in psychology where they show you the ink blots and you have to interpret it, however you want. And some people will say it's like a butterfly or a moth or, but basically we don't have a way of really classifying networks. We just kind of name them with some kind of gut feeling about what we think they represent based on our, knowledge of the literature. And so I think this particularly gets confusing in systems like this. This is a network, whatever you call it, it has nodes in the anterior insular cortices, the anterior cingulate, some frontal areas. But there's a bunch of networks that have nodes in those kinds of areas. In fact, there's one that Dosenbach calls the cingulopercular network. So that's including the anterior prefrontal, anterior insula, frontal operculum, dorsal anterior cingulate. There's another that Bill Seeley and I and others have called the salience network with nodes in the anterior insula, the dorsal anterior cingulate, subcortical structures. And there's yet another one that's been called often the ventral attention network by Corbetta and colleagues, which includes temporoparietal junction, ventral frontal, parts of the frontal cortices, and anterior insula. And so in this case, we see a few of these nodes are being represented across multiple networks. We also didn't get much agreement in the audience. We didn't get any agreement, I suppose, on what to call it. And I think we should be probably concerned. It's strange that we're not more concerned about these inconsistencies in the literature, because when you want to do a lit review or you want to understand what is all the previous literature in the brain that has to do with this, if it's schizophrenia, depression, whatever it is you study, you would want to have some principled way of figuring out if there's anything relevant to the thing you're studying. And I think it's hard when some of these same brain regions are popping up all over the place in different networks with different names that aren't standardized in any way, shape, or form. And so this is my favorite slide from this whole talk, because it just illustrates the point that if you were using different schemes or different atlases or different parcellations, you would really have a very non-precise understanding of what this network is. So even if you just said, I'm interested in every literature, everything out there on the salience network, different people call different things the salience network. So the Gordon parcellation, that's the one on the top left here, has a salience network that you can see there in purple. And then Thomas Yeo has a widely used 7-network parcellation that has something called the salience/ventral attention network. And as you can see, it's not quite overlapping with the Gordon. And as you keep going on, there's other parcellation schemes and other ways of dividing up brain cortices into different regions. And you can see that they call salience something very different again in the Shen parcellation. And then if you look again here at a different one, Shire 2012, they've got an anterior and posterior salience. So to me, it's very unsatisfying to be calling something the salience network and studying it, because whatever atlas you were referring to might have been using a whole different way of describing that same brain region. And so we were hoping to get, and for me, I guess I take it personally because I really care about those brain regions. My whole lab spends all their time studying the anterior insula and the anterior cingulate cortex. We have kind of been working for a long time with this idea that the anterior insular cortex in particular seems to act as a causal outflow hub, in the sense that if you use anything like Granger causal modeling or dynamic causal modeling and try to figure out, what brain regions are interacting and what the effective connectivity is between large scale networks, you can often predict what's going to happen later on. For example, in the frontoparietal cortex or in the default mode network from signals in the anterior insular cortex. So we think it kind of acts as a switch between some of these other networks in terms of directing neural resources. And that's all fine and good, except that we can't agree on what these other networks are, or what the salience network is even, for that matter. So part of this is that many brain regions serve multiple functions and are affiliated with multiple networks in a dynamic fashion. So it's not sort of as easy to map them one-to-one. So for example, the insular cortex is involved in processes like emotion, empathy, and pain, subjective awareness and interoception, all of these kind of, we think of as maybe more limbic kinds of processes, and somatosensation, but also in high-level cognitive processes and executive functions. So conflict, switching, attention, and inhibition almost always activate the insular cortex as well. So it's not that there's just kind of a one thing that this node might be involved in. And so there's a lot of complexity there. Another nice study from Thomas a while ago shows that if you look at all the kind of large body of task fMRI literature that's available in a meta-analytic way, and you try to break down sort of experiments into what tasks they're using and what cognitive components are engaged in each of those tasks, and try to map those onto voxels of the brain, you actually find that these areas in red are the ones that have the highest functional flexibility, meaning they show up in a variety of cognitive components and in a variety of different cognitive tasks. So they're basically multi-purpose kind of regions. And you can see in red here, the anterior insula and the ACC, very, very red parts of the brain, meaning they're functionally flexible. They're kind of going to show up in a lot of these different paradigms that people use when they use task fMRI. So again, maybe not surprising that the insula shows up in a lot of these large scale networks. It's also a very functionally diverse brain region. The dorsal anterior insula in particular, the one that's blue in this figure, shows a high degree of functional diversity. So there's a different study that I did in 2014 with Luis Pessoa and some collaborators, where we used meta-analytic connectivity modeling to ask questions about these different insula subregions and what other brain regions are coactive with them across multiple task fMRI studies. So all of these insular subregions are basically really kind of very active all the time across all different kinds of task paradigms. So it's hard to really isolate it to one network or one process because of this kind of multiplicity of function. And I'm sure, like many of you in this audience, are kind of experts on a lot of these methods, so I won't spend a lot of time on describing them, but feel free to ask questions if you're interested, especially since we're a small group, feel free to interrupt at any point. But you can do things like look at dynamic functional connectivity using sliding window analyses. Like in this case, you could take 45-second windows or 60-second windows of your longer resting state fMRI scan and create these whole brain correlation matrices, and then try to cluster the different windows into different states using things like k-means clustering. And you might find kind of changes from moment to moment in these whole brain connectivity patterns that I guess like Vince Calhoun calls it dynamic functional network connectivity. That's one way of thinking about it. So then you can quantify brain states like the frequency of occurrence of a particular whole brain functional connectivity pattern, or the dwell time, which is how long does that whole brain pattern persist once it starts, or state transitions, like how many switches between different states can you see? So these are all just ways of quantifying some of these functional brain dynamics. And when you do that with the insular cortex in particular, here at the top, there's just, a dorsal anterior subdivision, a ventral, and posterior seems to be split up into two. But what we try to do is look at how these insular areas talk to the rest of the brain in terms of their resting state dynamics. And what's interesting is that we found evidence for 5 different states, and some of the states, the different subregions are kind of looking not too different from each other in terms of their connectivity with the rest of the brain, like in state 3 here. And state 2, which occurs very infrequently, only 5% of the time, you see this divergence where the different insular subdivisions are actually talking to different parts of the brain. So just to summarize, if I can pop this back up, it's back. Okay. And if you look at the differences between the states, this kind of the panel on the left here just shows like how different each functional connectivity state is from each other functional connectivity state. And the dorsal anterior subdivision of the insula shows the biggest divergence or most differences between states in terms of its whole brain functional connectivity. The other subdivisions like middle, posterior, ventral are kind of not so different from one state to the next, but the dorsal anterior insula is much more functionally diverse. And that actually goes along with what we see in the task studies, where the greatest functional diversity in the task setting is also in the dorsal anterior insula. And it goes along very interestingly with a lot of these Granger and other causal analyses showing that the dorsal anterior insula, but not the other subdivisions, are actually the one kind of driving the default mode or the executive network. So anyway, insula is interesting and complex and fun, but it's just one example of why it's really hard to name and categorize large-scale brain networks, because the little bits of the brain that go into these larger networks have their own complex stories, and we have to account for all of these or try to make sense of them in our larger network nomenclature schemes. So these very fundamental existential questions that we're trying to deal with are: how is a functional brain network defined? And maybe how many functional brain networks are there, if we can even answer those questions, which I think we probably can't, but we're going to try. So the first attempt was, let me hide this. Well, maybe I can't. Oh, I got it. Yeah, these 3 dots. Hide. Okay. Yeah, thanks. Great. So Nathan Sprang and Thomas Yo and I tried to piece some of these ideas together in 2019 at some ski conference, and because, well, me and Thomas don't ski, but in any case, we had enough time to sort of sit down and think about these problems, about whether or not we could try to put together a universal taxonomy of these macro scale, kind of large scale functional human brain networks. So our first pass was to say, okay, let's forget about all these names you've heard, like salience and default and all the things that people like to use, and let's just go strictly with anatomy. And we'll say wherever in the brain that, you know, the network is encompassing, we'll use those anatomical terms to describe the network. So we tried to do that. Gosh, this thing really doesn't like my talk. It's so we tried to do that as a first pass, more as just kind of an exercise. So it'll come back eventually, it always does. So the first network, if you look on almost any parcellation scheme, or you look across independent component analysis, it's that first system we saw that everybody said, oh, it's a visual network, right? We often see this occipital cortical network covering the back of the brain. You see it in Yo's parcellation as a couple different parcels there in purple and red. You see it in Gordon parcellation. He calls it medial and lateral visual, the back of the brain. And if you look at like Smith, an old ICA paper comparing task and rest, you see a couple of networks there. But we thought, okay, at first pass, we could call something like that an occipital network, because it's, at the very least, it's in the occipital part of the brain. And then with motor stuff, it was also like, okay, motor, sensory, pericentral network. You've got the sensory cortices and the motor cortices right there around central sulcus. So if you look across, you know, resting state, ICA, task, ICA, 2 different, these are just for illustration, but a couple different, you know, parcellations. Blue is the somatomotor in Yo, and in the Gordon, it actually breaks down into more hand, face, foot, and premotor. Part of these, as you guys know, when you do parcellations, it's basically the clustering, other than saying we use this criteria to determine the optimal number of clusters. So you'd see oftentimes these some networks break down and further. In Yo, you've got a 7 network solution and a 17 network, for example. It's like, which one is correct? There is no way of really deciding, right? So this clustering issue leads to some of these problems that we're talking about as well. But so the, sensory visual stuff, we can kind of agree on the nomenclature, but when we get to like more high level association cortices, I think that's when our real problems emerge, because we don't agree. So here's what I'm calling just broadly a dorsal frontoparietal network. In Corbetta's kind of cognitive world, it's called the dorsal attention network, with nodes in the frontal eye fields and intra parietal sulcus. So that's sort of a classic dorsal attention network. It shows up in many kind of meta-analytic schemes. It shows up, this is really convenient. It shows up as green in both the Yo and the Gordon parcellation. And there seems to be some kind of overlap between these different dorsal frontal parietal networks across these schemes. But it starts to get even more kind of complicated with the lateral frontal parietal. So those are the dorsolateral prefrontal and posterior parietal areas that people often talk about with a bunch of different names. So central executive network, control network, multi-demand system. Basically, there's a lot of ways of talking about this system. It's in orange in Yo's parcellation, he calls it frontal parietal. And in Gordon's, it actually breaks down into a parietal memory and a frontal parietal, yellow and blue networks down here. It shows up a lot, like I said, in the Smith, it's kind of an executive network. And this, I'm showing here a meta-analysis of executive function studies, and it basically overlaps with what people call this frontal parietal network. But we're getting a little bit dicey in terms of the naming, right? Things are not quite standardized. This is the worst of all. So this is when you get to anything that includes the anterior insula and the dorsal anterior cingulate. I want to call it mid-cingulate insular network. Seeley started out calling it salience network. Corbetta, I think it overlaps a lot with his ventral attention network. It shows up in Thomas's work. He also did nice kind of seed analyses to show how different parts of the same network can also have like different connectivity patterns, illustrating some of this complexity. In Gordon, he actually has labels for all three of them within this parcellation. So ventral attention network is labeled separately from cingulopericular, which is labeled separately from salience. But if you remember in Yo's parcellation, ventral attention and salience were lumped together. And also it didn't overlap very well with the Gordon one anyway, but these are the problems. And then medial frontal parietal, which is the one everyone agrees is the default mode network. But still, like, what does default mean? Nothing. Like, it's a useless label. So like, why, if we're going to use useless labels, we might as well use anatomical labels that are more informative and say, okay, where in the brain does this network usually show up? It shows up pretty consistently, right? Every sort of parcellation scheme has a default mode network. Some people like to subdivide it into sort of medial temporal lobe and sort of a, with more core system and the more like peripheral system. So there, it does break down further, which is another kind of complexity to all this. But at the very first pass, we thought, let's just try to use anatomical terms and, try to move away from giving these functional names because people don't agree on the functional names, but it's possible they might agree on the anatomical taxonomy. But it turns out nobody really wanted to do this. So even though we pushed for it, nobody was willing to give up their salience networks and their, multiple demand networks or whatever they wanted to call them. So this was, when we wrote this paper, one of the reviewers said, why don't you form a bigger committee and get a bunch of people to agree and sit down and have a bunch of meetings about it? So I thought, oh, that sounds like a fine idea. But yeah, it didn't work, like I said, basically, the dynamics, the inter-individual variability, subcortical brain regions, fractionation, subsystems, none of the stuff I told you about was solved by the proposal that we made. But luckily we teamed up with Anastasia and others to come up with this really cool acronym, because the first thing you have to do in science is come up with an acronym, and then everything else falls out from there. So she came up with the work group for harmonized taxonomy of networks, WhatNet, right? So that was the first concrete step. Then we became an official organization for human brain mapping best practices committee. So are some of you familiar with the Committee on Best Practices and Data Analysis and Sharing, COBIDAS? That's the one that first started out trying to give standards for reporting fMRI analyses and results sections of papers. Was led by Tom Nichols, and there's a bunch of papers came out a few years back. There's another one on MEG and EEG by Ina Puce and Cyril Pernette. And then for some reason, the website says best practices on large scale brain network nomenclature, but really it's WhatNet. So we're a group for harmonized taxonomy of networks. So we were kind of trying to follow in the footsteps of best practices for data analysis and sharing and say, hey, can we all agree on these and start doing this in our papers so it'll make it easier going forward. So we met on Zoom every month starting in like pandemic times, I think it was like, maybe it was 2020. It was probably like right at the start of the pandemic when we started meeting on Zoom with all of these people listed in this paper. So it was like 26 of us. And it was like a lot of Zoom meetings during the pandemic and after. And we talked and we discussed all these issues, and we had structural people and we had people who, think networks or shouldn't be called networks. We had a philosopher. So we kind of took all of the different opinions, and a lot of people, some of these people created the atlases that we're talking about. So we tried to bring them together and say, hey, why can't we just, make some mappings between these atlases? There was a lot of talk. I think like 4 years went by. Right? I mean, this was a long process, but we did write a paper. Well, at the end of like the first 2 years, we decided we were not going to standardize the nomenclature. That was a hope that was dashed after, several years of these meetings. We had a lot of good discussions along the way. So we wrote like a really long paper describing all the controversies and progress and why there might be difficulty in coming to this standardization because of things like the dynamics, inter-individual, developmental differences, so many issues that are at present hard to overcome with some standard nomenclature. So we wrote about this just to get all our thoughts out. And then, well, one thing that came from this controversies and progress paper was that we did a survey like the one you guys did at the beginning of this talk, where we asked people, what do you think this network is? And just like this audience, the people of Twitter thought that it was very easy to name a motor network and pretty easy to name a visual network and pretty easy to name a default mode network. But if you got into anything with parietal or insula or frontal cortices, there was the least amount of agreement between raters. So anything that involved association cortices and any of the more complex areas of the brain, people did not agree what to call it. So we had, as kind of what you would expect, an agreement on some things, but real disagreement on others. Yeah, this was just, we put this in the paper to show, like, when you use any kind of thing to break down the brain signals, if it's ICA, your model order really affects how many networks you get. And so you can have subnetworks. And so, what would you call the subnetworks? This is one of the issues that we don't know how to overcome, right? And then the inter-individual variability, some people's networks are placed in slightly different parts of the brain. So how do you like map across them? These are from some of the Midnight Scan Club papers where they scanned an individual many times and got individual level maps. So, two of the people in our WhatNet group are like authors of these papers, and they were like really concerned about how do we capture these inter-individual variability issues. And then a bunch of people also brought up, well, we keep talking about these cortical parcellations. What about the basal ganglia? What about the cerebellum? What about thalamus? We're pretty, we kind of ignore a lot of that in some of these parcellations, which doesn't seem quite right, right? So this was our minimal reporting guidelines from the 2023 paper, where we, similar to like the COBITUS, here's a checklist of things you might want to think about. This was a very broad checklist that was basically saying, hey, when you, for example, when you think about a task fMRI contrast that you get in some paradigm, it's not necessarily in a GLM, univariate GLM, it's not necessarily going to correspond to any of these resting state large scale networks. So just kind of keep that in mind. And so we tried to encourage people to avoid labeling these patterns of brain activity with like a specific cognitive term, because that makes it harder for people to compare across studies. If you say, well, I'm using, I'm finding activation in the multiple demand network, and then someone else calls it executive control network, like it's impossible to do a lit search and find both of those things if you just use one term or the other. And so to determine network affiliations of novel findings, use and reference one or more existing parcellation schemes. So here's where number 3 is what I'll talk about in the next few slides, where we tried to make a tool to make this easier for people to do. And then we wanted to just keep in mind reporting variation from the population used to generate the reference parcellation scheme. So that means, so how many of you work in aging, like aging populations or Alzheimer's? Yeah, so older brains, do they look a lot like 35 to 50-year-olds? Not always. What about young? Like, does anyone do young development, like infant or 8 to 12-year-olds even? Yeah, so do they always look exactly like the adults? I mean, right. So I think, but do you often use the adult parcellation scheme to reference your findings? Yeah, because you have no other choice, right? So I think just reporting the sample variation, just saying we're using an adult parcellation scheme, but we know that things are different in aging and things are different in development. Just even acknowledging that, I think, is a first step. I'm about to write a grant to like do a bunch of parcellations for different ages, and we'll see if we ever get that grant. But I think we need like more, more parcellation schemes that aren't just adult parcellation schemes to really get at some of these questions. And then consider adding on, if you're using atlas labels from one of these well-known atlases, you could also use anatomical labels just to make it even easier for people to link up your findings with other existing findings in the field. And then, follow the COBITUS guidelines for connectivity. That's just more broadly referencing to the previous best practices guidelines. So number 3 here, the trying to deter, like if you have a new finding and you want to say, this is the default mode network, maybe people will agree with you, but if you want to say any of these other complicated parts of the brain, you want to label it, it's harder to decide just eyeballing it, right? So we tried to make this easier with this network correspondence toolbox. So this was really created by Ruby Kong working in Thomas Yo's lab. And it's what we try to do is make a Python toolbox that should be like out of the box. For me, like installing Python is harder than actually using the toolbox. So it should be easy once you've installed Python to get this to work. And if you have trouble using it, please email us and let Ruby know, because she's in the process of like developing it and making it even easier to use. But the idea is that you should be able to take whatever atlas and whatever parcellation scheme that you want and take your findings and say, okay, this is how much it overlaps with this network and this scheme, and do it across a bunch of atlases at once. So you don't have to just pick one. You can do it for 10 atlases. And you should be able to visualize that in a way that's, and quantify it. So you don't just eyeball it. You don't just guess, right? You actually, you have some way of coming up with numbers, right? So this is Ruby, and she spent the last year or two, probably 2 years now, developing this toolbox where basically data file is whatever your findings are. Let's say you just did some study and you want to create some labels for those activations, whether it's ICA, resting state task, it doesn't matter what it is really, but you can take your data file and you can select any, I think we have 16 atlases in the toolbox. So you can say, I want to compare it against all 16 atlases and find out what overlap is the highest. And so all this toolbox does is do a simple computation of spatial overlap between your activation or your connectivity, whatever that is. Compute the dice overlap across all the atlases and all the networks and all the atlases that you included. So it's just going to give you an output that's kind of like showing you the degree of overlap with each of the labels in each of the different networks. So here's this is actually, we did this in the beginning just to prove to ourselves what we sort of intuitively already knew, which was these different atlases use really different naming schemes that sometimes converge, but sometimes they don't. I think a good example from here is like, if you look at Yo's salience, ventral attention A from one parcellation scheme, that actually overlaps a lot with singulo-opercular from Gordon's parcellation scheme. So, they're really, and the salience over here in Gordon overlaps with salience ventral attention B in Thomas Yo's. So it's good to be aware of that, so you're not just like calling things whatever, without actually realizing that they might be the same or they might be different. And then if you take your own data, so let's say your data is the yellow map up there, and you kind of do a couple specifications like basically you just say which atlas list you want to compare it to. There's just 4 up there, but you could do all 16 if you wanted. And you could add your own atlas too if you want. So that's the other thing. So the toolbox will automatically compute this network correspondence using the dice coefficient and do these spin tests to give you some statistical significance values to say like which ones are like most correlated with your own map. So in this example, these yellow things up here are most correlated with frontoparietal in Glasser, control A and B in Yo, dorsal attention frontoparietal in Gordon, and frontoparietal in Chen. And then it gives you this plot over here, the radar, just to show you like another way of visualizing all of this. So I like it because you can see what all these different atlases are calling your activation, and then you can decide what to do with it as a user, whatever you want. But the point is now you don't have to do a lot of guesswork, and you don't have to say, well, it's this and this atlas and that atlas, because it's all visualized for you in one figure. Sometimes you get these like really easy activation maps, like this is a working memory contrast from Human Connectome Project. And in this case, like here, yeah, in Gordon, it's dorsal frontal parietal, but actually, wait, this one isn't that easy. But there, either way, like whatever you find, you can then report based on which significance level you decide you want to report on. I think this was the harder one. Yeah. So here we just show an example. We threw in some independent components from the UK Biobank data. So this is a little bit harder to label because it's kind of like all over the place, but you can still use this tool just to make some kind of inferences about what it most corresponds to. So I think it just basically makes your life easier. So this is the bioRxiv preprint that we put out, well, like 2 months ago, that has the code, it has the links to the GitHub and has like the toolbox in it. And a few people have started using it. If you find bugs, it's a great time to let us know, because obviously we want to make it usable for everyone. So this is up and it's under review and will come out someday, but why wait for that? Just, go ahead and go to GitHub and start using the tool, and do give us feedback if you think actually it'd be better if you displayed it this way, or actually, it'd be nice if you had this feature. We want to make this something that everyone uses if you want to use it. So feel free to provide feedback to any of these folks, especially, me and Thomas and Ruby. And, we'll try to make it a good tool for the field. So this is, oh yeah, that's it. Okay. So I'm happy to take any questions, and thanks everyone for coming out in the rain in the middle of August. Appreciate it.